The year was 1942, and the world was engulfed in flames. In military hospitals across North Africa, young men lay dying not from their wounds, but from something far more insidious: infection, gas gangrene, blood poisoning. These invisible enemies claimed more lives than bullets ever could. In the dim corridors of field hospitals, nurses moved between beds, changing dressings that reeked of decay, watching helplessly as healthy young soldiers deteriorated before their eyes. The smell of rotting flesh was everywhere. Morphine could dull the pain, but nothing could stop the relentless march of bacteria through damaged tissue. Lieutenant Colonel Hugh Cairns, a consultant surgeon with the British Army, had seen enough. In his tent in Tunisia, surrounded by casualties from the brutal desert campaign, he made a decision that would change the course of medical history. He had brought with him something precious, something still largely unknown to the military medical establishment. It was a yellowish powder, unstable, difficult to produce, and still considered experimental by most authorities. It was called penicillin. The story of how this miracle substance came to the battlefields of North Africa begins not in a laboratory, but with a stroke of extraordinary luck in a cluttered London research facility. Alexander Fleming, a Scottish bacteriologist working at St. Mary's Hospital, had returned from his summer holiday in 1928 to find something peculiar. A petri dish containing Staphylococcus bacteria had been contaminated by mold. But this was no ordinary contamination. Around the mold, the bacteria had simply stopped growing. There was a clear halo of death encircling the fuzzy growth. Fleming, with his characteristic scientific curiosity, didn't discard the contaminated dish. Instead, he studied it. The mold was identified as Penicillium notatum, and Fleming discovered that it produced a substance capable of destroying a wide range of harmful bacteria. He named this substance penicillin, but there was a problem. Fleming couldn't figure out how to produce penicillin in quantities large enough to treat human patients. The substance was incredibly unstable, breaking down within hours. After publishing his findings in 1929, Fleming's discovery gathered dust for more than a decade. The scientific community showed little interest. The paper was largely ignored. Fleming himself moved on to other research. Meanwhile, in the laboratories of Oxford University, a team of researchers was about to resurrect Fleming's forgotten discovery. Howard Florey, an Australian pathologist, Ernst Chain, a German Jewish refugee, and Norman Heatley, a British biochemist, began investigating penicillin in 1939. What they found astonished them. In laboratory tests, penicillin destroyed bacteria without harming human cells. It was precisely the kind of weapon doctors had dreamed of for centuries, a way to fight infection from the inside. But producing penicillin remained an enormous challenge. The Oxford team resorted to desperate measures. They grew the mold in every conceivable container: bedpans, milk churns, food tins, even bathtubs. The laboratory became a bizarre penicillin factory with 6 women, who would later be known as the Penicillin Girls, tending to the fermenting broth day and night. Ruth Callow, Claire Inayat, Betty Cook, Peggy Gardner, Megan Lancaster, and Patricia McKegney worked tirelessly, farming precious milligrams of penicillin from gallons of mold broth. It was painstaking, exhausting work, but it yielded enough penicillin for the first human trials. In February 1941, an Oxford policeman became the first person to receive penicillin treatment. He had developed a severe infection with abscesses throughout his body. Nothing conventional medicine could offer had helped. He was dying. Within 24 hours of receiving penicillin, his condition improved dramatically. The abscesses began to heal. His fever dropped. For the first time in weeks, there was hope. But the supply ran out. Despite attempts to recover and purify penicillin from the patient's urine, there simply wasn't enough. The policeman died a few weeks later. The team was devastated, but they had seen enough. Penicillin worked. They just needed to produce it in larger quantities. Florey and Heatley knew that British pharmaceutical companies, already stretched thin by the war effort, couldn't help them scale up production. So in July 1941, they did something extraordinary. They traveled to the United States, carrying with them samples of the precious mold. But they feared German spies might intercept them, so they rubbed penicillium spores into the linings of their coats. If they were captured, if their samples were destroyed, they could still restart the entire process from the mold growing in their clothing. In Peoria, Illinois, they found allies at the United States Department of Agriculture's Northern Regional Research Laboratory. The American researchers had expertise in fermentation techniques that the British lacked. They developed deep fermentation tanks that dramatically increased penicillin yields. They discovered that adding corn steep liquor, a waste product from corn starch production, created an ideal environment for mold growth. Production increased exponentially. But they needed an even better strain of penicillium, so began a global search for mold samples. Soil samples arrived from around the world, from military bases in remote jungles and deserts. Scientists tested thousands of specimens, looking for strains that produced more penicillin. Then, in a twist of fate that seemed almost too perfect to be true, a laboratory assistant named Mary Hunt found the winning strain at a local market in Peoria. It was growing on a moldy cantaloupe. This strain, eventually designated as Penicillium chrysogenum, produced more than 200 times the penicillin of Fleming's original mold. By 1942, American pharmaceutical companies began gearing up for mass production, but production was still limited and every dose was precious. The military had first claim on all supplies. Civilians would have to wait. On March 14, 1942, the first soldier received penicillin treatment for blood poisoning. He recovered completely. The medical community began to take notice. Then came the case that changed everything. Ann Miller, a 33-year-old woman in New Haven, Connecticut, lay dying in March 1942. She had developed a streptococcal infection following a complication related to pregnancy. Her temperature spiked past 106 degrees. She drifted in and out of delirium. Doctors tried everything, sulfa drugs, blood transfusions. Nothing worked. Her husband, through a connection to a professor at Yale who knew Florey's team, managed to obtain a small amount of penicillin. It was rushed to the hospital. The transformation was miraculous. Within hours of the first injection, Ann's fever began to drop. Her blood cultures showed the bacteria count plummeting from hundreds of colonies per milliliter to just one. She made a full recovery and would live for another 57 years. News of her recovery spread rapidly. Suddenly, everyone wanted penicillin. But the war took priority. In early 1943, at Bushnell General Hospital in Brigham, Utah, military doctors faced a crisis. They were treating more than 200 war-related orthopedic patients. Most had compound fractures, bones shattered and protruding through skin and muscle, and most had developed osteomyelitis, a severe bone infection that sulfa drugs couldn't touch. These men faced a grim choice: amputation or slow, agonizing decline toward eventual passing. The hospital requested penicillin for what they called an intent-to-treat program. It wasn't a formal clinical trial. There was no time for the niceties of proper scientific protocol. These were wounded soldiers, and they were running out of options. The penicillin was administered both by injection and applied directly to wounds. The results exceeded all expectations. Infections that had resisted every other treatment began to clear up. Bones began to heal. Men who had been told they would lose arms or legs kept their limbs. Word spread quickly through military medical channels. Meanwhile, in North Africa, the real test of penicillin's potential was about to begin. Howard Florey himself, along with his colleague Hugh Cairns, traveled with the British Army to Tunisia in 1943. They brought with them precious supplies of penicillin. Cairns, the consultant surgeon, had seen the devastating effects of infection on wounded soldiers. He knew that in previous wars, more men had perished from infections than from their actual wounds. He was determined to change that. The fighting in North Africa presented unique challenges. The desert environment meant wounds were immediately contaminated with sand and dirt. The intense heat accelerated bacterial growth. Traditional antiseptic methods often failed. By the time wounded soldiers reached field hospitals, infections were already well established. Many had gangrene setting in. The standard treatment was amputation, often of multiple limbs. Cairns and Florey began treating casualties with penicillin, but they also made a radical proposal. Instead of waiting for infections to develop and then treating them, what if they prevented infections in the first place? What if they gave penicillin immediately after injury, before bacteria could gain a foothold? This was revolutionary thinking. Most military doctors had been trained to debride wounds extensively, removing dead tissue, and then leave wounds open to drain. The fear of gas gangrene was so strong that no one wanted to seal up a wound that might harbor deadly bacteria, but Cairns argued that with penicillin, they could clean wounds thoroughly and then stitch them closed. The penicillin would handle any residual bacteria. He demonstrated this approach on fresh casualties arriving from Sicily in July 1943. Soldiers who would have faced weeks of painful open wound treatment instead had their wounds closed within hours. They received regular penicillin injections, and they healed, cleanly, quickly, without the massive infections that had plagued combat casualties for centuries. The transformation was so dramatic that surgeons began changing their entire approach to battlefield trauma. One British surgeon with the 21st Army Group described being struck by the remarkable well-being of soldiers treated with penicillin. Their wounds were painless. They showed none of the signs of systemic infection that had been considered inevitable. Surgical teams that had been performing 30 or 40 wound closures a day found their work accelerated. No longer did they need to spend hours debriding and packing wounds. They could clean, close, and move on to the next patient. But supplies remained critically limited. Penicillin was so valuable that medical teams routinely recovered it from patients' urine, purified it, and used it again. Every drop mattered, and the stakes were about to get much higher. Operation Overlord, the D-Day invasion of Normandy, was being planned for June 1944. Thousands of casualties were expected. The success or failure of the invasion might hinge on how quickly wounded soldiers could be returned to fighting strength. The United States government made penicillin production a top priority. 21 pharmaceutical companies worked together in an unprecedented collaboration. Pfizer converted an old ice factory in Brooklyn into a penicillin production facility with 14 massive fermentation tanks, each holding 7,000 gallons. By March 1944, just 18 months after the BBC had reminded listeners that good science takes time, this single factory was pouring out penicillin by the ton. By June 6, 1944, when Allied forces stormed the beaches at Normandy, they carried with them approximately 2.3 million doses of penicillin. It was enough to treat the initial wave of casualties, and it made an enormous difference. Between D-Day and VE Day, the 21st Army Medical Group recorded approximately 406,000 admissions, including 182,000 battle casualties. Of these, at least 100,000 received penicillin treatment. The majority were wounded men who, in any previous war, would have faced amputation or would have succumbed to their infections. The impact on amputation rates was profound. During World War I, between 12 and 15% of wounded treated in frontline hospitals perished from infections, most notably from gas gangrene. With penicillin, that rate dropped to just 3%. Vascular injuries, which in World War I had necessitated amputation in nearly 50% of cases, saw amputation rates drop to just over 20%. Soldiers were keeping their limbs. They were surviving what should have been fatal infections. And they were doing it because of a moldy piece of bread that Fleming hadn't thrown away 16 years earlier. But the story of penicillin in World War II isn't just about statistics. It's about individual soldiers whose lives were transformed. Lieutenant Colonel S.W. Thompson, a Seaforth Highlander, was wounded in the thigh during fighting in Sicily in 1943. He became one of the first field trial subjects for penicillin, treated by Fleming himself and a colleague. Thompson received the drug in powder form, applied directly into his wound. Within 5 weeks, he was back on the front lines. In a previous war, his injury would have meant months of hospitalization at best, amputation at worst. Stuart Hastings Bull was blinded and severely wounded in Normandy. Infection set in. He was isolated alone with his pain and fear, certain he was going to pass away, but penicillin pulled him back from the brink. When he finally emerged from isolation, a fellow soldier called out in surprise that Bull wasn't deceased after all. The miracle drug had given him a second chance at life. The psychological impact was as important as the physical benefits. Soldiers who had seen their comrades lose limbs to infection or perish from wounds that seemed minor knew that penicillin offered them hope. They called it their magic powder. Almost to a man, soldiers reported that they believed penicillin had saved their lives. This confidence, this belief in their medical support, made them better fighters. They knew that if they were wounded, modern medicine stood a chance of bringing them home whole. The ethical questions surrounding penicillin use were complex and troubling. Because supplies were so limited in the early years, difficult decisions had to be made about who received treatment. Military casualties took priority over civilians. But even among military patients, choices had to be made. Should penicillin go to the most seriously wounded, those fighting for their lives? Or should it go to those with moderate injuries who could be returned to duty quickly? There was no good answer. One particularly contentious debate involved the treatment of soldiers with certain intimate infections. Penicillin was extraordinarily effective against these conditions, curing in days what previously took months to treat. But should precious penicillin supplies be used on soldiers who had contracted these infections through their own choices, when other soldiers with combat wounds were waiting for treatment? Military medical officers argued passionately on both sides. Eventually, pragmatism won out. A soldier cured of such an infection could return to duty in days. A soldier disabled by such an infection might be out of action for months, reducing fighting strength. Both groups received treatment. The success of penicillin on the battlefield drove even faster development. Chemists worked frantically to determine the exact molecular structure of penicillin. If they could synthesize it chemically, production could increase exponentially. Dorothy Crowfoot, a brilliant crystallographer at Oxford, took on the challenge. Using X-ray crystallography, she painstakingly worked out penicillin's structure. It was groundbreaking work that would eventually earn her a Nobel Prize. But chemical synthesis remained elusive. It wouldn't be achieved until the late 1950s, long after the war had ended. Meanwhile, pharmaceutical companies refined production techniques. They discovered that different strains of penicillium produced slightly different variants of penicillin, each with its own properties. The British researchers, using surface fermentation, primarily produced what came to be known as penicillin F. American companies using deep fermentation produced penicillin G. Both were effective, but had slightly different applications. This diversity would eventually lead to the development of dozens of different penicillin derivatives, each optimized for specific types of infections. In the Pacific Theater, American medical teams faced challenges even more severe than those in Europe. The tropical environment was a breeding ground for bacteria. Wounds became infected within hours in the oppressive heat and humidity. Soldiers fighting in the jungles of New Guinea and the Philippines faced not just enemy fire, but dengue fever, malaria, dysentery, and a host of other tropical diseases. Compound fractures in this environment were particularly dangerous. Bone infections developed rapidly and were notoriously difficult to treat. American doctors began using penicillin not just for battle wounds, but for the myriad infections that plagued troops in the tropics. They treated soft tissue wounds, blood infections, and pneumonia. They even used it preventatively, administering penicillin to soldiers before operations to prevent post-surgical infections. The results were remarkable. Soldiers who should have perished instead recovered. Operations that should have failed instead succeeded. But even as penicillin saved thousands of lives, medical teams learned its limitations. Some bacteria were resistant from the start. Others developed resistance after repeated exposure. Doctors learned that penicillin was useless against certain types of infections. They learned that dosage mattered enormously. Too little penicillin and the bacteria survived, potentially developing resistance. Too much and the drug might be wasted on a single patient when it could have treated several. The nursing staff played a crucial role that often went unrecognized. In field hospitals and evacuation centers, nurses managed the complex logistics of penicillin administration. The drug had to be given by injection every 3 hours around the clock. Nurses maintained rigorous schedules, ensuring that patients received their doses on time. They monitored for allergic reactions, which could be severe. They managed the recovery and purification of penicillin from urine, a distasteful but necessary task when supplies were limited. These nurses worked under conditions that would have broken many people. They treated soldiers with horrific wounds in tents that offered little protection from the elements. They worked impossibly long shifts, sometimes 48 hours or more, when casualties were heavy. They dealt with the emotional trauma of losing patients despite their best efforts and the joy of seeing men they'd thought were doomed walk out of the hospital on their own legs. Many of these nurses pushed boundaries when they felt it was necessary to save lives. When official protocols said to wait for approval before administering penicillin, some nurses, seeing a soldier deteriorating rapidly, made executive decisions. They knew they might face disciplinary action, but they also knew that delay could mean the difference between life and the loss of a limb or worse. These acts of medical courage rarely made it into official records, but they saved lives nonetheless. There were failures, too. Some soldiers received penicillin too late. By the time they reached medical care, infection had spread beyond the point where penicillin could help. Amputations were still necessary. Some soldiers still passed away from their wounds despite receiving the best care available. These losses weighed heavily on medical staff. They had this miracle drug, this incredible tool, but it wasn't a panacea. It couldn't undo catastrophic damage. It couldn't reverse tissue death that had progressed too far. The experience with penicillin also taught military medicine valuable lessons about logistics and supply chains. Getting penicillin from factories in the United States to field hospitals in Europe and the Pacific required careful planning. The drug was unstable. It needed to be kept cool. It degraded rapidly in tropical heat. Special packaging was developed. Refrigeration units were installed in medical facilities. Supply officers learned to account for losses to spoilage and damage in transit. By the end of the war, the system for distributing medical supplies, particularly penicillin, had become highly sophisticated. As 1945 dawned, penicillin production had reached unprecedented levels. What had been precious drops in 1941 was now being produced by the ton. By the time of the German surrender in May 1945, penicillin was no longer in critical shortage for military use. Enough was being produced that civilian populations in Allied countries could finally access the drug. By 1946, penicillin became available to the British public. The age of antibiotics had arrived. The impact of penicillin extended far beyond treating battlefield wounds. The war had created an enormous infrastructure for producing antibiotics. Pharmaceutical companies had invested heavily in fermentation technology. They had trained thousands of workers in sterile technique and quality control. They had developed distribution networks that could deliver temperature-sensitive medications across the globe. When the war ended, this infrastructure didn't disappear. It was repurposed for peacetime medicine. The success of penicillin inspired researchers to search for other antibiotics. If a mold could produce a substance that killed bacteria, what else might be out there? Soil samples from around the world were tested. New antibiotics were discovered: streptomycin, which could treat tuberculosis, chloramphenicol, tetracycline. Each new discovery built on the foundation laid by penicillin research during the war. But perhaps the most important legacy of wartime penicillin use was the proof of concept it provided for collaborative scientific research. The Manhattan Project gets most of the attention when people discuss World War II scientific achievements, but the Penicillin Project was in many ways more remarkable. It involved cooperation between British and American scientists. It required government agencies, universities, and private companies to work together. It demanded the sharing of proprietary information and research data. It showed that when faced with a common enemy, whether that enemy was fascism or bacterial infection, science could achieve the seemingly impossible. The story of penicillin development also highlighted the role of women in scientific advancement. From the penicillin girls tending the mold cultures at Oxford to Dorothy Crowfoot solving the molecular structure to the countless nurses administering the drug in field hospitals, women were integral to every stage of penicillin's journey, from laboratory curiosity to battlefield miracle. Yet their contributions were often minimized or forgotten in historical accounts that focused on famous male scientists. The soldiers who benefited from penicillin carried its legacy forward. They returned home with limbs that previous generations would have lost. They built families that previous generations would never have had the chance to create. They contributed to the post-war economic boom with bodies that could work, with minds that weren't consumed by chronic pain from untreated infections. The ripple effects of penicillin's impact on World War II extend through generations. Modern medicine owes an enormous debt to the wartime development of penicillin. The techniques developed for mass production of antibiotics formed the basis for manufacturing countless other medications. The clinical trial methodologies refined during penicillin research established standards that are still used today. The understanding that bacterial infections could be treated systemically from inside the body opened new avenues of research that continue to bear fruit. But the story of penicillin also carries warnings. Even during World War II, some bacteria showed resistance to penicillin. Today, antibiotic resistance is a global crisis. The bacteria have adapted faster than we've developed new antibiotics. Many of the infections that penicillin once cured easily now require much more powerful and more toxic antibiotics. Some infections have become resistant to nearly every antibiotic available. We face a future where common infections might once again become life-threatening. The mechanisms that made wartime penicillin development possible, the collaboration between government, academia, and industry, the willingness to share research openly, have been replaced by a system that prioritizes profit over public health. Pharmaceutical companies have largely abandoned antibiotic research because antibiotics are less profitable than medications for chronic conditions that people take for years. The soldiers of World War II understood viscerally what we risk forgetting. That infection is an existential threat. They knew comrades who perished from infected scratches, from wounds that seemed trivial, until bacteria turned them into something deadly. They understood that the ability to fight infection is not something to be taken for granted. It is a hard-won achievement of human ingenuity and cooperation. As we look back on the development of penicillin during World War II, we see a story of scientific triumph. But we also see a story of human courage and compassion. Researchers who worked themselves to exhaustion. Nurses who held the hands of dying soldiers and refused to give up. Doctors who made impossible choices about who would receive limited supplies of a miracle drug. Soldiers who endured agonizing wounds and experimental treatments with the hope that their suffering might help those who came after. The preserved limbs, the saved lives, the returned soldiers. These are the true measure of penicillin's impact. Every man who walked off a hospital ship on his own 2 legs instead of being carried off on a stretcher. Every soldier who kept his arm and could hold his child for the first time. Every family that didn't receive a telegram informing them of loss. These are the victories that penicillin made possible. The nurses and doctors who administered penicillin, often under the most challenging conditions imaginable, deserve recognition equal to that given to the scientists who developed it. They were the ones who translated laboratory research into saved lives. They were the ones who sat with soldiers through long nights, monitoring for complications, adjusting dosages, fighting infection, one patient at a time. They were the ones who decided, sometimes in violation of official protocols, that a soldier's life was worth the risk of using experimental treatments. The forbidden aspects of early penicillin trials weren't about rogue individuals acting recklessly. They were about medical professionals who recognized that traditional protocols designed for peacetime conditions might condemn soldiers to needless suffering or loss. When the choice was between following regulations that would guarantee amputation or trying an experimental treatment that might save a limb, many medical professionals chose compassion over compliance. History has vindicated their choices. Today, we take antibiotics for granted. We expect infections to clear up with a few pills. We consider it a minor inconvenience to take medication for a week. The soldiers of World War II knew better. They understood that infection was a death sentence. When penicillin gave them a reprieve, they recognized it for the miracle it was. Their gratitude, their amazement at being given a second chance, should humble us. The development of penicillin reminds us that progress in science and medicine doesn't happen in isolation. It requires resources, cooperation, and a willingness to take risks. It requires people who are willing to push boundaries, to question established procedures, to try something new even when success isn't guaranteed. It requires societies that value human life enough to invest in its preservation. As we face modern challenges, from antibiotic resistance to new infectious diseases, we would do well to remember the lessons of wartime penicillin development. When faced with a crisis, collaboration works better than competition. Open sharing of research accelerates progress. Diverse teams, including people from different backgrounds, genders, and nationalities, produce better results than homogeneous groups. And sometimes the most important breakthroughs come from the most unexpected places: a moldy cantaloupe in a Peoria market, a contaminated petri dish in a London laboratory. These humble origins gave rise to one of humanity's greatest medical achievements. They remind us that discovery often comes from careful observation of the world around us, from recognizing that something unusual might be important, from following curiosity wherever it leads. The soldiers whose limbs were saved by penicillin rarely knew the full story of how this miracle drug came to be. They didn't know about Fleming's accidental discovery, about the penicillin girls working in Oxford laboratories, about the frantic efforts to scale up production before D-Day. They just knew that the yellow powder sprinkled into their wounds, the injections they received every few hours, these things were keeping them alive, keeping them whole. That was enough. For those medical professionals who pushed boundaries to use penicillin when official protocols said to wait, the vindication came in the form of healed wounds and preserved limbs. When a soldier they'd treated with unauthorized penicillin walked out of the hospital on his own legs, when a man they'd thought might lose his arm instead regained full function, that was all the justification they needed. They had taken a risk, and it had paid off in the currency that mattered most: human life and human dignity. The story of penicillin in World War II is ultimately a story about hope. Hope that human ingenuity can solve even the most intractable problems. Hope that cooperation can overcome obstacles that defeat individual effort. Hope that compassion and courage can change outcomes even when the situation seems hopeless. And hope that what seems impossible today might become commonplace tomorrow if we're willing to do the hard work of making it happen. As we conclude this journey through the development and deployment of penicillin during World War II, we're left with both gratitude and responsibility. Gratitude for the researchers, medical professionals, and soldiers who proved that bacterial infection need not be a death sentence. Responsibility to preserve and extend their achievements, to ensure that future generations benefit from effective antibiotics, and to remember that medical miracles don't happen by themselves. They happen because people care enough to make them happen. The thousands of soldiers who kept their limbs because of penicillin lived full lives. They became fathers, grandfathers, teachers, engineers, artists. They built the post-war world with hands that might have been lost, walked into the future on legs that might have been amputated. Their lives stand as testament to what medical science can achieve when the stakes are high enough, when the commitment is strong enough, and when human compassion guides human ingenuity. In the end, the story of how one nurse's forbidden penicillin trial saved thousands of soldiers' limbs is really the story of how many nurses, many doctors, many researchers, and many ordinary people working in extraordinary circumstances came together to achieve something remarkable. It's a story of scientific brilliance and human courage, of desperation and hope, of failure and triumph. It's a story that reminds us of the best of what humanity can be when we face our worst challenges. And it's a story that continues to resonate today as we confront new threats to human health and seek new solutions. The legacy of penicillin extends far beyond the soldiers it saved in World War II. It transformed medicine. It ushered in the antibiotic era. It proved that humanity could fight back against the microorganisms that had plagued us since the dawn of time. And it showed that when we work together, when we share knowledge freely, when we put human welfare above personal glory or profit, we can achieve miracles that change the world. That is the true story of how penicillin saved thousands of soldiers' limbs, and it is a story worth remembering.